The drug is absorbed mostly through the cell membrane, which serves as the cell's outer barrier, in which drugs must be able to pass through. In crossing the barrier, there are various mechanisms in which drugs can pass through. It can be through passive diffusion for water and lipid-soluble drugs, or facilitated diffusion, which is also known as active transport, or it may be through endocytosis. The type of mechanism used by the drug is determined by the size, water, solubility, and lipid solubility of the drug. When it comes to passive diffusion, the concentration gradient across a membrane separating two bodily compartments provides the driving force for passive drug absorption. In other words, the substance goes from a high concentration area to a low concentration area. Passive diffusion does not involve a carrier that is not saturable and shows a low structural specificity. This mechanism is responsible for the absorption of the majority of drugs. Water-soluble medications pass through aqueous channels or holes in the cell membrane, whereas lipid-soluble medications can easily pass across most biological membranes due to their solubility in the lipid bilayers. On the other hand, in facilitated diffusion, additional substances can enter the cell via specific transmembrane carrier proteins that help large molecules move through. These carrier proteins undergo conformational changes, allowing drugs or endogenous chemicals to penetrate into the insides of the cell and move from a high concentration area to a low concentration one. Moreover, this type of mechanism is not energy dependent, but it can be saturated and can be hindered by chemicals that compete for the carrier. Now we will move on to active transport. Specific carrier proteins that traverse the membrane are also involved in this route of drug entry. Using these specialized carrier proteins, a few drugs that closely mirror the structure of naturally occurring metabolites are actively carried across the cell membranes. The hydrolysis of adenosine triphosphate drives energy-dependent active transport. It is capable of transporting pharmaceuticals along a concentration gradient from a low drug concentration region to a higher drug concentration region. This process is saturable. Active transports are selective, and other co-transported chemicals can suppress them competitively. Next type of transport mechanism is through endocytosis. This kind of absorption is utilized to transfer extremely large molecule drugs over the cell membrane. Endocytosis is the process of a medication being absorbed by the cell membrane and then being transported inside a cell by squeezing the drug-filled vesicles. Endocytosis transports vitamin B12 across the gut wall, whereas exocytosis releases neurotransmitters such as norepinephrine, which are then held in intercellular vesicles at the nerve terminal.